for open and reproducible research, subtitled, How Do I Share Clean Data? So first we're going to start off with everybody's favorite, a quiz, like quizzes, right? So how many people here practice open science or think that they practice open science? No one? No one does anything that they think would be open. Okay, well maybe I'll change your mind by when I tell you, so what is open science? So we can take a very simple definition. This is a relatively recent way of sort of describing what it means to be open. And it's understood by this easy acronym called FAIR. So open data, open science are things that are findable, accessible, interoperable, and reproducible. So both for humans, both for people, biologists, other people, consuming it, but also for our computer overlords, right, that we're, feeding data into and getting results out of. So what does it mean to be findable? That means that you can type in a URL and you'll always get to that piece of data, regardless of how much time has passed. Similar for accessible, that there are certain restrictions or maybe no restrictions for accessing it. Maybe you have to fill out a form that says you're going to use it in a certain way. But there are a set of standards for ways to access it. It's interoperable, so I can give it to you, you can give it to somebody else, and they can all work on it in pretty much the same kinds of ways, fairly easily. And it's reproducible, meaning that if you give me the set of steps that you've done on something, and you give me those steps and the things to do it, then I should be able to go through the steps and get exactly the same result as you do. So we'll talk about this maybe a little bit later, about the difference between the use of the term reproducible and the use of the term replication. So reproducible really just means that I do the exact same things that you do and I get the same result. That doesn't tell me that I can replicate your experiment, meaning I do your experiment again with new samples, with new data, and get the same results, in the statistical or the biological sense. But for our purposes, reproducible just means I can do exactly what you did and I get the same result. There's a really good recent paper that sort of codified this. So this is the work of a working group called Force 11, I think, that was started by Phil Bourne, who is now the head of all data management, basically at the NIH, the big data to knowledge thing. And they just released a paper in Science that in scientific data that sort of codifies these standards and trying to push this out to more people. So that's sort of just a very, one definition of what open research means. But why? So why do we do things like this? And again, I'm not going to rant too much about this because this is coming from our work at Sage, where we're trying to make things more accessible to people. So if we're open and reproducible, we allow people to others to build on our work and transfer knowledge. We're trying to improve transparency and discourage fraudulent work, though there's no magic bullet for that. If somebody wants to fake their work, they're going to fake their work, whether they're transparent or not. They'll just work harder at it. So that's not an answer. Another good reason is the funding agency tells you to. And so this is becoming increasingly common. And this is a link to the NIH's bevy of data sharing policies for different groups and different grant types that you may or may not be applying to. And they're getting more strict about what kinds of data you have to share and when you have to share it. So good thing to know about that. It can help you find collaborators. So if you're sharing things out there, people can find you on the internet easily. You might actually find people to work with you. One of the biggest barriers that some people think is that you won't get credit for it somehow, right? So we're in this system where publications are the currency that people's careers often sort of depend on in some kinds of ways. But it turns out that if you have data, it doesn't have to be big data. It can be the data that you're working on in your lab, the data that you're working on in a particular paper. You can get credit for that in terms of a publication or other metrics. And so I'll share these slides on Synapse, and I'll share them with you. These are just 4 journals that I know of that you can publish papers about the data you generate. You don't have to define any hypotheses. You don't have to have any scientific breakthrough to get that new science cover paper, right? But just the act of describing your data and making it available to people is something that you can get credit for. And I think that's one of the big incentives for people to becoming more open with the data that they have and show that they're not just somebody's not just toiling away and then they have to wait for the paper to come out in order to actually share it with people. So why not open and reproducible? And these are a few of the things that we hear. It's risky for early career researchers, right? So you're giving away your data. What am I going to do with it? I can give a great counterpoint, and this is a really interesting guy. His name is Dr. Titus Brown. He's an evolutionary biologist at MSU. He made it in his all of his statements, like research use statements, said he was going to be open from the very beginning. He signs all of his reviews of papers. He publishes all of his primary author grants. No one is scooping him. No one is stealing his ideas. No one is getting to things before him. Ideas are a dime a dozen. People have their own stuff. It's really not, I think, as big of a problem as people think. There are publisher limitations, right? So some publishers, there are certain ways that they want you to go about releasing your data. They often embargo it, but then they're also making money off of you. That's a whole other discussion about open access. We're not going to get into that. I don't know if anybody has heard or seen the research parasites meme that went around. So New England Journal of Medicine published a very divisive paper about data sharing in clinical trials and basically said that if you're out sharing your data with people, they're just going to take it and run away with it and never talk to you again, and they're just parasites. So it's this very divisive topic of polarizing topic that, I mentioned that you don't get credit. Well, there are ways that you can get credit for it. I can talk about some of those other ones. Sort of in my view, getting scooped is if somebody is out there taking your work that you're doing, there's a word for that. It's called plagiarism. So if you use a preprint, if you put your data out there, you maybe need a license, some licensing on it, and there can be share alike. There are lots of ways to get a license, and there's a nifty site here that lets you choose a license. It's choosealicense.com. If somebody is taking that work and deriving it and then passing it off without giving you credit for it, that's called plagiarism. And so there are ways around that. So I don't think that everyone needs to be scared about sharing data and getting scooped, but there are always caveats, and there's always reasons to do something and not to do something. But how do we do this? So I'm going to give you just some general advice, some general concepts, and then talk about one segment of this very specifically. So it's not easy, right? I mean, it takes a lot of time, and that is actually one of the biggest barriers to do this. It really changes the way that you work and you organize, and if you only have a limited amount of time, there's only so much you can do. I guess in my mind, being partially reproducible is better than not at all being reproducible. So you have to pick places where you can make improvements and then just do that. And so your next paper, maybe it's putting the data that you use to generate your figures in a file that can be downloaded and somebody else could regenerate your figures and making that part of your supplementary data. That is a step towards making your work reproducible. That's kind of a simple thing that a lot of people is sort of tractable. So the three major steps: plan before you start, and this applies to anything. I don't need to tell you this. Organize and keep track of things, archive and share. I can point you to a good article called Baby Steps for the Open Curious. Really good place where it tells you kind of, things you can do here and there that can, increase your open impact, so to speak. But I'm just going to really talk about data sharing best practices. So how do you share data, either with someone who is analyzing it, like a bioinformaticist like me, or these things really apply to if you're sharing it with the public or with a larger consortium like the PCBC. You're putting it on Synapse or you're putting it on the web somewhere. These are some of the things that I think are important. And this does not necessarily apply to big data. You don't have to go out and do 500 RNA-seq experiments and that. It could be you did a PCR experiment on some cell lines and you want to be able to share that so somebody else can use it, right? I mean, the scale does not need to be big data per se. So this thing says click me, so I'm going to click it. So this is actually the pretty much same talk that I gave for this course in Bozeman. And I actually stole almost all these materials from not stole, because they were openly available, so I borrowed them from this very prominent researcher. Yeah, I'm a parasite. Who is actually here sometime soon, I think maybe even this week, Dr. Jeff Leake, very prominent statistician, bioinformatician, and also an advocate for education in the open access and open data sphere. So who is the target audience? Are scientific collaborators who need someone to analyze data for them. Maybe you're a student or a postdoc looking for consulting advice, and which is probably the case for A lot of people here who are working with core. Yeah, do you have a question?
I was wondering, we all know the importance of data sharing.
Mm-hmm.
Can you tell us like a list of things, open shared platforms that
I will get there. Yes, definitely.
and
Mm-hmm.
also maybe tell us like how we could use Amazon Cloud to share or store,
Mm-hmm.
because we all know this philosophy, but we
Mm-hmm.
don't know the tools
Yep.
for it.
Mm-hmm.
Yeah.
Okay, yeah, definitely. Well, that will be something we cover. I will make a point to cover here. So you know who the target audience is. Basically, I just want to avoid some pitfalls and sources of delay, the constant emailing back and forth that you're going to have to do with a statistician or a computational analysis, computational person. Can kind of skip this. So it's hard, but make friends with your data scientist or your analyst, because we like making your data better. You know, we like working with clean data and helping, and we want to help you, and it actually helps us when we can help you and we can reduce this back and forth time. So just ask. I already kind of said why. So what are the 4 things that you should be delivering to the analyst? Raw data, a summarized, tidy data set, and I'll talk about what that means. A code book, or data dictionary as it's called, describing the variables and the values that are in your summarized data. And then the steps that you use to take number 1 and go to steps 2 and 3. So the processing that you did to go from 1 to 2 to 3. And depending on what you're doing and who you're working with, the burden of who does those steps might shift from you to them. But if you do anything, then we want to know. So what is the raw data? The strange binary file that you get out of the PCR machine. So Bio-Rad spit out this PCRD file and probably a bunch of other things. Some Excel file with a bunch of worksheets that some company you contracted sent you to get some results. Maybe even just hand-entered numbers that you collected looking through a microscope or counting something on Petri dishes on plates or something like that. So that's raw data. You didn't do anything else to it. How do you know if it's raw? You didn't run any software. You didn't change any of the numbers. You didn't remove anything from the data set. And you didn't do any summarization. So you haven't grouped anything together and calculated a mean or a median, or said, oh, I don't like this one. Let me just delete that row from my spreadsheet. Things like that. So that's the raw data. So what's the code book? So for almost any data set, the measurements you calculate have to be described in more detail than you can fit into a spreadsheet. So the spreadsheet is not the place to tell me what the units of measurement are for each of your columns, or what machine you use to run them on, or how you made in your summary statistics, in your summary type data set, how you decided to make choices about summarizing, or information about how you designed your experiment. Not a good place to put that. So that goes in a separate file. But what goes into that file? So there are basically a finite number of variable types that most people have. So continuous numbers, positive infinity, negative infinity, ordinal numbers, categorical things, dates, things that refer to other objects like gene names, gene symbols, mouse strains, things like that. Missing data and censored data. So we'll talk about each one of these. So how do you code continuous variables? So continuous variables, anything measured on quantitative scale. That could be any fractional number. So what are some examples? Gene expression level, concentration, weights of mice, for example. So again, the units go in the code book, not in the data. So your column should just have the values, and then the code book should tell me that these are weights in kilograms, for example, so that I know the scale. That'll become more apparent tomorrow, or maybe later today, depending on how far we get. If we're putting things like 10 kg in every cell, and then we want to do some mathematical manipulations on that, that makes it difficult in R or another programming language to do, because what does 10 kg plus 5 kg mean to a computer? Computers don't have any idea of what kg stands for other than it's a string of a k and a g, right? So it has no concept of units. We need a number 10 that's a number plus a number 5 in order to do some kind of mathematical analysis on that. So how do we code ordinal data? So ordinal are a fixed small number of ordinal levels. So for example, survey responses, poor, good, fair, quality control levels, pass, fail. Better not to convert continuous variables to ordinal ones in your data. So if you have some way that you can take continuous and bin them, for example, so high, medium, low, You don't want to convert them. You just maybe want to add another column or describe what your criteria for high, medium, and low were. From 0 to 5 was low, from 5 to 10 was medium, for example. So that's placed in the code book for it. Categorical data are unordered multiple level data. So male or female, treated, untreated, control, sample types, different cell lines, for example, or your drug names that you're using, for example. What do I have here? This is not terribly important. Dates. Dates are always a tricky thing. And if you ever worked and I'm sure everyone here has worked in Excel, and dates are something that get changed around all the time in Excel, depending on which version you're using, whether it's on a Mac or Windows. But they are important. So the ordering matters. So for example, 3/11/2015 is not the same as 11/3/2015, depending on which country you're in. Different people use different ordering of month, day, and year. Even the difference between putting a 4-digit versus a 2-digit year can be problematic for some software, and they won't know what to do with that. They'll think 15 means the year 15, not the obvious 2015, right? Also be careful about text columns that can be interpreted as dates. Everybody's favorite gene OCT4. If you type that into a cell in Excel, it will convert it to 10/4/1900 or 10/4/1901, depending on which version of Excel you're using. So always be very careful in Excel or copying and pasting things. If you search OCT4 in databases like Geo, or search the year, you'll find tons of supplementary data spreadsheets with 10/4/1900 in them. It's disappointing and problematic, but kind of humorous. Missing data is one of the bane of computational people, because what does missing mean? So this is a great place for things that have to be in the codebook, and this is probably one of the biggest things that we deal with all the time, is what does it mean to be a missing value? So first of all, missing data are missing, and you don't necessarily know why they're missing. You don't know. But the most important thing is to code them in a consistent way. So for us, NA with no quotes around it, N/A, a blank string, or an empty space, to us, these are all different things, and they could have a different meaning. And when we put them into a computer, they don't necessarily all get treated the same. So consistency is the key, and explain which value you're using and maybe why. Anything you know about why data might be missing is essential for computational analysis. But then there's censored data, right? So censored data is you know the missingness mechanism of some level, right? You had an equipment failure. A patient lost to follow-up because they dropped out of a clinical trial. That's the common censoring. Some measurement being below a detection limit. So you got a value, but you have no confidence in that's the value because the machine can't read that low, for example. So these are censored data. If you don't have it, then you should code it as NA, but otherwise you need to tell an analyst that it's censored somehow, and the value is there, but we can't really do anything with it. So how do you censor for example, in a spreadsheet? You add a new column, and you say what variable you're measuring, censored, and then if it's true, then that value should be censored, and if not, then it's false. Right? That's one way to do it. There are other ways. That's the easiest way for us, I think, to add that data in. So entity names. So this comes up a lot in computational biology. Whenever possible, you want to use standardized entity names. So these are genes, proteins, reagents, etc. And when in doubt, you should check with a trusted naming consortium on how to name things. So most people here are probably working in human or mouse. There are very specific controlled vocabularies for the names of genes. And so the example is what POU5F1 is OCT4, right? OCT4 is actually not the standardized gene name, so any place you actually use OCT4, most computer programs will, some of them are smart enough and can figure it out, but it's hard, and don't know what that is. So you'll do a match and say, oh, here, put all these genes into this software package that's doing enrichment, you put OCT4 in there. It doesn't actually include it because it looks it up, and it's like, I don't know what OCT4 is, because the official gene symbol for OCT4 is POU5F1. All right, so these are important caveats, and it's probably the greatest unsolved problem in computational biology is mapping from one gene name or gene symbol to another name or gene symbol. There's so many databases, so many identifiers, right? So I think I had that in my thing, right? So for some for example, the Entrez ID for POU5F1 is 5460. Nathan showed this earlier. It's some number, chronological number. There's a transcript ID, so if you're working and you know specific transcripts, then you should use the transcript ID. You could use it from here, from RefSec, as long as it's from a trusted place that records all of this information. If you're working with proteins, POU5F1 isn't particularly useful. You should use the SwissProt ID, so to refer to the actual protein instead of just the gene. For example, the Sigma-Aldrich monoclonal antibody for POU5F1 is P0082. So these are useful identifiers that just in tracking and going back and making sure that the right things were used are useful for us and to differentiate between things. So if you use 4 different antibodies for POU5F1 because you were testing them, then you should probably record where you got those in a standardized way so that we could test the difference between them. And as Larson mentioned before, you may have heard this, reproducibility is kind of a big deal, blah, blah, blah. So when you submit a paper, right, everyone should be able to replicate your analyses. And most biologists I say most because I think everyone works in good faith, are good about releasing the biological steps that they do, but often shun the computational steps, partly because they don't know how to explain it, or the person they're working with whose job is to explain it didn't do a very good job, so somebody like me who's not doing a very good job. And it's hard, right? So it's hard to know what to explain. Sometimes you're limited by word count, and then you want to stuff it somewhere in supplemental, and then somebody, whatever. So things happen, and then nobody does it. So there are lots of reasons. But you should strive to making things, as described as possible, even from the computational side. And as you're getting into these genomic level analyses, lots of parameters, lots of different programs to use. So it's really important to be as verbose as possible, even to be wordy in the sense of this. And sometimes that's what goes into one of these scientific data paper, like a data paper that's just describing your data. You can actually write an entire another paper and get credit for it and cite it anytime you use this data set. And that's where some of that information can go instead of not putting it into the biological paper that you're trying to write. And this includes I think the biggest thing, and it's hard for us, is how do you reproduce figures? And hopefully we'll get to tomorrow to show you how in R you can make publication quality figures automatically. No more Excel, dragging and dropping, messing with colors. Like everything can be very programmatic, high resolution, grouped graphics, all sorts of crazy stuff. So hopefully we'll get to that and be able to show you some basic steps that you can do to make scatter plots, heat maps, box plots, all the kinds of things that people use. Even if you can't make if you publish a paper and you can't do these steps to make your figure available, then the best thing to do is make the data available that you use to generate the figure, the Excel spreadsheet or the CSV file or whatever, so that somebody else can go and be like and make that figure again, and then maybe subset it in a different way or look at it in a different way, analyze it with some different statistical method, for example. I'll skip that. So this is not a one-sided thing, right? If you're working with an analyst. So if you're sharing with the public, it's a little more one-sided because they're just going to be consuming. But if you have a consultant or a bioinformatic analyst or a core that you're working with, you should expect some things back from them. They should give you an analysis script that performs each of the analysis, not just some instructions that you're going to have to Google in order to understand everything, right? An actual script and then instructions of how to use it and what the assumptions were, et cetera. The actual code that they use to run it, maybe it's on an Amazon instance, maybe it's on your local compute cluster. How do they actually go about running it? And then any of the output files, figures, et cetera, that they generated, hopefully using the techniques that I described to make it as reproducible as possible. And there are ways that they can make it so you can, with some rudimentary knowledge of R and some GUI tools, get the thing that they did and start to run it yourself. You may struggle with it for a while, but you should get something back from them that you could use or give to someone else if you, end your relationship with them and somebody else comes in. So, blah, 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 I'll share that with you later. Oh, I just said all of that. So if you're not getting this, ask. And if you're still not getting it, you should break up with them. You should find somebody else. Hopefully you have the ability to find somebody else to do this kind of analysis. But these are things that you should be expecting from someone else who is doing your analysis. If they're not giving that to you, then it's not fair. You're doing all this work. If they're asking you, sending you emails, what does this mean, blah, 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 and then they're not giving you value added back, then you should find someone else. Then there is someone else out there. There are more people like us than you think. And you may have to go. Yes, it is. There's plenty of fish, right? Yeah. Yes, please go away. So, and then we have to talk about sharing, and this is what you're getting to. So the one place, since you are all in the PCBC, hopefully you've at least seen the Synapse website at least one time in your life. And you may have thought that it was only a place where PCBC data was going and it was being disseminated and there's this wiki and some other stuff. But it's actually a tool that you can use freely available to upload data, to share it with people, to get citations for it, to do attribution, a lot of things. So let me just actually switch back. I think I'm actually have that. Okay, so review. There's a bunch of links in here that give much more details about the things that I was talking about. So consistency, saving data and playing. I actually didn't talk about some of these, unfortunately. So let me run through a couple of these. Since I have a few minutes. I have till 12, right? Oh, cool. So I talked about consistency, doing the same things the same way, NA values in the same way, et cetera, et cetera. We like plain text. So even Excel files, they look like plain text, but they're not. You send it to me. I can't just open it in a text editor and look at it. It's going to look like a big jumbled mess that I can't look at. So we want, and from Excel, you can export to CSV, tab separated, save as text, sometimes it's called, I'm not sure. That's the best way to share data with a wider audience because it's the most interoperable. I can open it in any editor and at least look at it and see that things that make sense, that there are numbers in it and not gobbledygook, or that there are characters, that there are strings in a particular column, for example. You get a very easy visual representation. And you can always go the other way, so any Excel or spreadsheet software can read a CSV, TSV, and open it in Excel for you to poke around and take a look at it. I hate on Excel a lot. It has uses, but it also has abuses. So there are definitely many uses for it, and I use spreadsheet software on a regular basis as well. So, but in terms of sharing data, it's not always the best way. Your data should essentially fit in a rectangle. So if you can't turn your data into a rectangle of rows and columns, then spreadsheets are never going to be the answer. So you have to think of something else, and you may have to work with a data scientist. Sometimes you may need multiple rectangles of data. So it's maybe you're describing clinical samples about clinical features about a set of patients, and then you're taking measurements on those patients multiple times. Those kinds of things don't necessarily go in the same thing because you're going to have a bunch of stuff repeated over and over again. For example, you don't need to write the gender of the patient every time you take a measurement because you could store that in a table about patients. So not super important, but sometimes it makes sense to break your data up into multiple rectangular discrete things, but and then we can always merge them back together. We can always put them back together. Take care with date to discuss that, data dictionary, and then mechanisms for actually sharing it. So let's actually get to that. So sharing with Synapse, as I was mentioning, freely available to you and actually to anyone. You can go and start your own projects there. We meet all of these FAIR criteria. We make anything you put into Synapse findable, accessible. It's actually up to you to make it interoperable, so following the things that I just noted, and reusable, sort of up to you. We are not the only ones, so I will give you some other ones here. OSF, Open Science Framework, is another one. Data Dryad is a very common one that you can upload pretty large data sets. Figshare is nice, so you can you can both store data and figures that you've generated and get citations for them, DOIs for them as well. Really useful. GitHub is another good place for it. I'm sure people have heard of GitHub or other source code repositories. It can work some for data, but it can work well for analysis files. So we use it a lot to share versioned analysis files that are then often cited in papers, for example. One nice thing about Figshare is you can take a GitHub repository that may have some code. You can version the entire thing, save it in Figshare, and make it citable that way. So there are lots of ways that you can share both data and code. So I'm going to talk about Synapse because it's our product, and I can talk to you with most of the features. So the kinds of things that you can do with Synapse: organize your research assets. So we have file storage, unlimited file storage, file size, number of files. It's unlimited. We do offer some structured data. So if you need to make a table, you need to do some kind of queries on it, like select where value is greater than 5. We have a powerful query system to have structured data. So both in terms of getting credit, so we record provenance. So you start with some input file and you generate some other output file. We provide a mechanism to show that there was an activity performed that links these two together. It shows your name. It shows when you did it, for example. So you can give these things as, hey, I actually did something to this data, and I can prove it because here's the record that said I did it. You can get DOIs, so document object identifiers. These are things that can immediately be citable in a paper. So you can get a DOI and you can cite a whole project, a specific data file, or any sort of level in between. And then we offer things for collaboration. So obviously, projects start as private. You can work them as your own private workspace, and as you want to share it with more people, you can start adding individual people, or you can just make the entire thing public. And it's very granular. You can share specific files. You can share specific folders. You can share the entire thing with any number of individuals or a larger population. And lastly, we have extensive features for documenting what you've done. So basically, they're all wiki pages, so they're collaborative editing. So anybody who has access that you've given, write and edit access can go in and write about a particular file, a folder about the project that they're working on, add new pages, for example, that's documenting what it is people are looking at, how they can use it, for example. So just to summarize, data storage, it's free. Data sharing, private, public, somewhere in between. We give documentation, provenance, which is tracking the steps to generate different data files, attribution, hey, I did something, I can tell somebody else about it, and citations. And as a caveat, there are other places provide these as well. I know OSF, Open Science Framework, is another one that provides a number of these things. We're not really competing in this space, but it's good to know that other things do exist, and Synapse is not the only solution or necessarily the best solution with what you're trying to do, but it is a good solution, and we can provide you with help on it. So, oh, thank you. Yeah, sure.
Is there any provision for protected health information? Even like a
You can put human, but you can't put any identifiable information, and most places are like that. You got to go to dbGaP or one of those if you're gonna. Actually, I don't think dbGaP has identifiable information. That's a whole other realm of legal and ethical conundrums that we're not involved in. You can put human data, but it has to be de-identified on Synapse. Yeah.
Well, the people are even saying that now that there's more question about something, even like a SNP is like identifiable even if it's de-identified.
Yeah. So I mean, there are ways that you can restrict it further. So, for example, you can make things private or public, but you can also add that somebody has to sign something to say that they're not going to de-identify someone. So you can add a barrier in there that says, I'm not going to allow you access to this until you fill out this form and email it to me, and I add you to this list of people. And now I have a record that says you agreed to something particular, and now I can hold it to you legally, for example. So we have some other ways of control access, other than just who you share it with, who you decide can see it. They have to consent to something as well. Yeah. Anything that's public in Synapse is subject to auditing, and so we allow also flagging. So anybody who's browsing around can say, oh, hey, this is personally identifiable information. You click a little button that flags it. Somebody from our governance team takes a look at it essentially as soon as possible, and then decides whether that should be there or not. If you are working in a private project, we don't audit anything that's not being shared with anyone. So If it's private, then we don't look at it. We can never look at it at all. But if it's public and somebody flags it, then we can say, this is something that's suspect. So for example, like that, if a paper comes out that says that used that SNP array and they have de-identified someone, we might want to think about whether that should be there or not. Yeah. So other questions? So we'll hopefully, are we doing a Synapse? We never did a Synapse. Yeah, okay, so to answer your question more completely about how you can use the tool that we have, Larson's going to show how we use it in the context of the PCBC, and hopefully you'll get an idea of how to use it there. But we can talk offline, too, about some of the other options, and I'm happy to talk with anyone about my experience with some of the other options like Figshare. So I've used Figshare twice for 2 papers. So I wrote a bunch of analysis code and packaged it up on GitHub, and it was a citation for me in the paper. So I was second author on the paper. We made all of our figures. I have a file that generates the supplementary information and all the figures in the main text, and I can cite that in the paper and say the code that generated all of this data is here. Here's all the analyses. It wasn't easy. I'm not going to lie. It takes a significant amount of work, but like I said, baby steps. Maybe just making the raw data that you had available to people in a publicly available place is a great place to start with things. So, yeah. Cool. So after lunch, we're going to talk about R. Yeah.
So you guys work with people from different universities to maybe publish, so they ask you guys for help with some of their analyses, and then you help them with some of your codes, whatnot, and analyze the data, and then they cite you as paper. Can it be different universities or only to your university?
Are you speaking as Sage as a company, like helping people with using Synapse, or just in general?
Like, for example, you were talking about somebody was using your code to do a paper and you became a second author. So was it with Synapse or?
No, actually, so that was actually before I started here at Sage. So I was doing that completely independently. I could have gone back and turned my project into a Synapse project. But our data and everything else was also in another public repository. It was microarray data, so it's in Geo. But in our code, we link to Geo so that anybody can run it, get the data from Geo, rerun all the analyses, do all the statistics, generate all the figures and everything. But the thing that I created is citable separate from the paper we published. So I cited it in the paper, but then on my CV, I can say, see this citation of this thing I created that necessarily wasn't the actual analysis we did, but I did a lot of work, running, getting the microarrays ready to run and doing the statistical analysis for them and curating all the data. And those are things that you should get credit for, which often you don't get credit in the paper other than acknowledgments. KD did data analysis, right? You're a byline in a paper. So I have a way to show that I actually did a lot of work in other stuff. So. And Synapse can provide similar things as many of these other tools can too. So, yeah.
So we actually there's lunch in the back. We don't start the R session until 12:30. So if someone needs help who wasn't here yesterday to set up with the compute.
And let me put